Okay, I'll just start sharing my screen. And meanwhile, please feel free to introduce yourself in the chat box. There might be participants from our previous workshops or coursework. It'll be good to know that you have joined. Or if you're joining with us for the very first time as a part of this webinar, please feel free to introduce. It is always good to see fresh faces. You can also let us know a little bit about your background. Are you from a life science background or are you coming from a tech background who's looking to transition to bioinformatics and NGS? It'll be good to know all of these. Okay, so we have started receiving few responses in the chat box. So we have Anchal, who is pursuing masters in computational biology and bioinformatics. That is good to know. I would request the participants to unmute themselves and yeah, thank you so much. If there's anything, please feel free to let us know in the chat box and we'll be happy to take up your queries or your messages. Thank you so much, Anshul, for introducing yourself and welcome to our community. Okay, Pushkarni is doing BTech Biotechnology from DY Patil University, Pune. Welcome to the webinar. Okay, meanwhile, the participants are introducing themselves. I'll slowly start with introduction to the webinar. And so in the first part, I'll just introduce you to the team and some of the study resources available to you, and also an exciting announcement about our upcoming coursework. And then I'll pass it over to the mentor to start with his session. And once this part is over, if the participants have any questions, please feel free to post them. And for all participants who have joined with us just right now, we have an exciting announcement at the end of the webinar where we'll be announcing some exclusive discounts available for participants to enroll in one of our coursework. So please stay tuned till the end of the webinar. So without further delay, I'll start with the recording. And meanwhile, please feel free to introduce yourself in the chat box. We are always happy to see your introductions as well. Okay, so hello everyone, a very warm greeting to one and all, and I would like to welcome you all for today's webinar on Next Generation Sequencing Laboratory. And in this webinar, we'll be exploring the evolving world of NGS and its growing relevance in research and industry careers. Now, before we begin, let me also take a brief moment to introduce you to the team behind the webinar. I can see that there are a few participants who are joining with us for the first time. So for them, we are a global bioinformatics and data science company with roots in India and the US. And over the years, we have had the privilege to collaborate and partner with esteemed universities and industries across the globe. And our mission is to bridge the gap between data-driven research and skill development. So to achieve this, we work with regional and international experts who help us tailor our programs to the specific needs of students, educators, and researchers across the globe. Now, with the growing demand for skilled bioinformaticians and data scientists, we have developed a wide range of training and research offerings, including online courses, workshops, internships, fellowships, training programs, research services, and even a degree program as well. Now, here on the screen, you can see a quick overview of the student learning roadmap. Now, it is suitable for all participants of all levels, even from beginner as well as advanced level. So it takes you from a beginner that is starting with basics of bioinformatics. Following, you will be specializing in specific omics field like genomics, transcriptomics, metagenomics, and so on. And for those who are looking to go a little bit beyond this, we also conduct a research program where participants get the opportunity to work on real world research projects under the guidance of mentors as well. Okay, so I'll briefly cover those in the upcoming slides as well for those participants who might be interested to work on research project. Now, here's a comprehensive collection of our courses and example projects on a diverse range of topics on genomics, metagenomics, transcriptomics, as well as several case studies based on different topics like precision medicine, agri-bioinformatics, and oncology, infectious disease, neuroscience, and so on as well. So at the end of my presentation, I'll take you to our OmicsLogic portal where all of these coursework are available. And for those participants who are interested to explore, I'll just give you a quick demonstration of how you can navigate and find these. So all of these coursework, they're enriched with engaging content, including visuals, infographics, video lectures, and we also heavily focus on experiential learning through short tutorials, practical demonstrations, and coding exercises. And while our coursework are fully automated and lesson-based, we also understand that some participants might need the guidance of a mentor while they are studying. So that is why we also conduct workshops and training programs that combine live mentor-guided lectures by walking them through different topics so that as to help reinforce several of the concepts that participants might find it hard to understand. So this also includes a combination of expert video lectures, interactive tutorials, and step-by-step lessons. And these are all curated to ensure that learners are able to get both, best of both, that is the theoretical as well as the practical application. Now, here are some of the few of the many feedback that we have received from the participants across our training programs and workshops. Now, here's a quick announcement that I was talking about in the beginning of the webinar, that we're excited to launch a brand new 6-week online course on sample to sequencer, a practical guide to NGS workflow. So this course will be led by Dr. Rahul Ramakar, who's a seasoned clinical genomics expert with over a decade of hands-on experience in NGS, single-cell research, and translational genomics, and who's also the speaker for today's session. And over 6 weeks, participants will be exploring several of the key components of sequencing pipeline, including NGS platforms and their applications, sample preparation essential, library preparation techniques, and we will also cover several other important concepts like experimental design, targeted sequencing approaches, and real-world case studies. Now, to ensure an engaging and effective learning experience, this course will also include live mentor-guided sessions, supplementary reading materials, as well as a certificate of completion that you can showcase on your resume and on your LinkedIn profile. Okay, so we'll be covering more about this towards the end of the webinar as well. So if there are any specific questions related to the coursework, I'll be happy to address those as well. Now, this was the research program I was talking about in the beginning of the webinar. So we often get a lot of questions from the participants who are interested to work on research project. So this is a research program where participants get the opportunity to work alongside a mentor, and they can choose to specialize in a specific omics field like genomics, metagenomics, transcriptomics, cheminformatics, or metabolomics, and work on a specific topic of their choice like infectious disease, agriculture, precision medicine, metabolic disorders, and so much more. And I would like to quickly walk you through some of the ways that students from our previous research programs, how they have leveraged the various study materials to achieve their academic and professional goals, hoping that this might also inspire you to achieve the same. So here on the screen, you can see a cohort of students from Systems Medicine graduate program from Georgetown University who worked on machine learning projects using Python. Students also joined the research program so that they can present their research project later on at research symposium and poster presentation competitions to win exciting cash prizes. Students also join to get a research publication done that gives them this extra competitive edge over other students while applying for higher studies or in the job market. And faculties from universities have also collaborated to carry out publication work as well. And for those participants with a knack for science and business, we also offer an entrepreneurship and business innovation track as well. So now, with that, I'll just quickly take you to the OmicsLogic platform where all of the study materials and some of the ways that students have leveraged the resources will be available for you to view as well. And then I'll pass it over to the mentor. So for those of you who are joining with us for the first time, I'll encourage you to head over to this link, that is omicslogic.com website. And for those participants who have been previously part of our workshops or training programs, or even our coursework, you may already have an account on the portal. So you can just please pause for a couple of seconds till others sign up on the platform as well. Okay, so this is the learning management system that we use for providing all of these training as well as research related study materials. So once you head over to this link, you'll be able to see a similar interface. And to create an account, you can click on the login option and click on create an account. And you can enter your name, email ID, provide your password, and you can create an account. Or if you already have an existing social media handle, such as Google, Facebook, Apple account, GitHub account, or even Twitter, you can use any of these social media handles as well, and you can log in. Okay. Now, for those participants who have previously created an account on the platform, just enter your existing email ID, password, and you can log in. In case you're not sure, you forgot your password to your account, no problem. Just enter the email ID that is at least associated with the account, and then just click on forgot password, and you will receive an email in your inbox for resetting your account. Okay. So with that, I will log into my account using my Google ID. Now, once you log in, the very first interface that you will be seeing is our courses page. So this is the main course library collection that we have on several of the topics that I was showing you on my slides as well. And there might be a lot of participants who might be coming from a life science background, biotech background, pharmacology, microbiology, and so on, who might be new to the field of bioinformatics. So if you are someone who's interested to explore this field, there's an excellent coursework that is Course 1 Introduction to Bioinformatics that you can explore. So you can just click on course 1 and you will be able to see. So here you will be able to see several of the lessons that are part of this coursework, and this will introduce you to what is next generation sequencing, what are the various omics data types, how they are generated, and how they're used in healthcare. So you can explore this coursework as well. Now, for those participants who might already be well equipped in bioinformatics skills, then if you're interested to specialize in a specific omics field like genomics, transcriptomics, or if you are interested to sharpen your coding skills, there is Python, R, as well as there are also coursework on machine learning and data science and several other omics fields as well, like metagenomics, single cell transcriptomics, epigenomics, and we also have some technical coursework like Linux and GitHub as well. So that is one way you can explore some of our coursework on our platform. But apart from that, if there are any specific keywords that you're interested to explore, for example, let's say transcriptomics, you can simply type in and you can see a comprehensive collection of coursework and case studies that is related to the field of transcriptomics. And you can see case studies related to a diverse range of topics like infectious disease, neuroscience, and so on as well. I'll just quickly open up one of the coursework that is transcriptomics that is quite popular amongst the participants and most liked by the participants as well. So this is the format. I just want to show you the format of the coursework just so that you can have a glimpse into it. And I'll just open up one of the lessons, and all of these lessons contain visual content. There are interactive materials, there are short tutorials within each of these lessons as well to help reinforce some of the concepts. And there are also assignments, quizzes to test your knowledge at the end of each of the lessons as well. Okay. So this is just a quick introduction that I wanted to give you about some of our resources on the platform. But if there are any specific questions related to some of this collection of coursework, we'll be happy to take it up towards the end of the webinar. Okay, so with that, I will stop sharing my screen and I would like to invite the speaker for today's session, Dr. Rahul Ramekar, to share his insights on the topic. Over to you, sir.
Thank you. Thank you very much, Shri Gauri. Hold on. Okay. Good evening, everybody. Happy Teachers' Day to all of you. And thank you. Thank you very much for taking out the time today. So today's topic we are discussing is breaking into multi-omics and why next generation sequencing matters. As Shri Gauri has also explained that we have designed a course. So also, of course, this webinar is also related to the course where we are going to give the information, but it also explains why we have designed this course, why it is important for all of you, because you are interested in next generation sequencing, to learn the nitty-gritties of next generation sequencing. NGS as a cutting edge technology, right? So what we'll start with one case study first. Okay, now picture this, or imagine that a newborn baby girl, so just a few days old. Okay, so she's having seizures after seizures. Okay, and because of those seizures, she's not able to feed on her mother. Okay, her brain scan, it was inconclusive. Okay, she had done routine blood test, completely normal, and yet, even after few minutes, her body starts shaking because of those seizures. And for her survival, feeding tubes are important. Okay, so doctors are doing everything what they know, but again they are left guessing. Okay. Now imagine that being her parent or her doctor. Okay, so you're doing everything right, but nothing is working. Okay, and here every hour counts, because that can save her life. Okay, so this is what the a true story of Sebastiana. So she's from US, and actually what changed everything was a simple blood test. Okay, what she did was a rapid whole genome sequencing. Okay. So with help of those rapid whole genome sequencing, the doctors were able to identify certain variations in one of the gene, which is very important for, let's say, for that ion transport gate channel, when most of you have learned in our cell biology basics, right? So what happened was there is a single rarest of rare mutation happened in that particular gene, and because of that the gates or the ion channel was open, and there was continuous influx and outflux of the ions, the proton gradients. Right? Because of that, she was getting seizures. Now, before that, before going for the whole genome sequencing, they tried everything, but it did not work. Once they got the results, the conclusive results, immediately doctor changed her medications. The approved, FDA approved drugs were given, and she's fine, well, okay, and now she's in her teens. Okay. Now, the why this case study was discussed is because we should ask few questions. We are all students of science, right? So these are the basic questions in biology. Why is it that two people who are exposed to exact same virus, I'm talking about COVID, okay, so they had completely different outcomes. Okay. So what happened when we were infected with COVID? Few of us we ended up in ICU. Some of us we did not even react. It was plain like a regular cold and cough for us. Okay. So again, why do two patients with let's say same cancer takes same drug, yet one of them responds while other sees no benefit? In not only in humans, when we are talking about plants, the crop species, right? So we have seen that few of the varieties, okay, which are the hybrid varieties, they withstand drought or even pests, while the others, it requires care. They can't withstand that drought, heat, cold, stress. Okay? And perhaps the most importantly, how do we analyze, or how do we understand the individual building blocks of disease, so that we can predict, prevent, or even reverse them. So these are not just medical questions. So these are the puzzles where we need to answer them, of course. But how can we answer? It's like understanding the genetic variation. Okay, so some of the variations are inherited, passed down, like let's say biological blueprints. Other arise spontaneously, but together they determine how our body behave, or how our bodies behave. So how we grow, fight infections, or even metabolize drugs, and also how we age. Okay. So, of course, genetic variations is not that complete answer, but this is a starting point, right? So many of you might have question like, what is genetic variation? Okay, let's take a step back. Okay, so before addressing what is genetic variations, let's understand what exactly is DNA. So think DNA as an instruction manual for life. Okay, and for them it is the four letters, the four building blocks: adenine, guanine, cytosine, and thymine, ATGCs, are very important. Okay, so when we compare the DNA of two people, or let's say we compare between two populations, though these letters, okay, AGCTs, aren't always identical. Sometimes there is, let's say, a single letter is either deleted, added, swapped, okay, or there are chunks of those letters that are missing, or also swap. Okay, so that difference is what we call as genetic variation. And these variations are what makes each of us unique. Okay, so what they do is they influence simple things such as your hair color, skin tone, or let's say, I mean, how certain how we how the drugs affect us. Okay, so when these variation occur within a gene, they can create slightly different version of that gene, and we call it as alleles. Okay, and alleles are the reason why you might have, some of you might have brown eyes, some of you might have blue eyes, of course black eyes are there. So in short, genetic variation is, let's say, nature's way of writing millions of tiny edits into the instruction manual, making sure that, let's say, no individuals are exactly same. And so the big question, what we actually asked initially, that why, okay, and how genetic variations affect. So another question is, how do these tiny variations or tiny changes at, let's say, at DNA level leads to such a big difference in health or appearance or diseases, right? So to understand that, you should also understand what the central dogma of life is. I'm pretty sure you all know that, right? So it's how the genetic information flows from DNA to RNA to protein. Right? So DNA is the blueprint, the master plan. RNA is the messenger, right? And also carrying those instructions. And proteins are the workhorses, right? They do all the building, repairing wherever our body it needs. Okay. Now, when a variation occurs at, let's say, at DNA level, okay, that can ripple or travel through the change of those central dogma, right? Sometimes it also changes the RNA and eventually protein. Okay, but of course, it's not that, it's also not that simple, or whether we are in that central dogma, right? So there is another layer, that is layer of epigenetics. Right? It decides which genes are turned on or off and how strongly. So this is why, two people with the same variant may still have very outcomes depending on how that gene is expressed. So of course, when I'm talking about those genetic variations which ripple through those central dogma, right? So it is not that universal formula where every DNA mutation will affect each layer. Okay, biology is much more complicated than this. So, for example, at, let's say, at the protein level, okay, so in protein level itself, there are a lot of post-translational modifications that can drastically change how protein behaves, and therefore the final phenotype. Okay, so but of course, this the understanding the genetic variations at a different level gives us, nevertheless, it gives us the framework. Why should we, and also it gives us a starting point to answer our questions. Now, so understanding genetic variation at, let's say, now we'll talk about, hold on, let me, yeah. So let's say we are talking about genetic variations at DNA level. Okay, so I mean, it helps us to understand the most fundamental questions. Let's say we are talking in terms of clinical genomics, whether it's the variations are affecting or whether the variations are in let's say in cancer, they are inherited conditions, right? A single mutation in a tumor, let's say a suppressor gene or mismatch in a hereditary gene can be a difference between, let's say risk and reassurance. While in microbial species, the understanding genetic variation at DNA level, I mean, the microbiome, right? So they helps us to understand how the microbes are interacting with our body, right? How they are influencing health, immunity, or even crop productivity. Okay, and of course, there are also few complex questions like whether the is the chromatin open or closed? Is a promoter methylated? So DNA, I mean, it's not just a linear code. It's a structurally dynamic. Okay. So, for example, I will give you the breast cancer, right? So think about, so in breast cancer, there are two important genes, the BRCA1 and BRCA2. And in those genes, there are certain mutations, right? So those mutations, when they might be inherited, okay, most of them, there are inherited variants, which dramatically increase a woman's lifetime risk of breast and ovarian cancer. Now, by understanding the variations, okay, at DNA level, so we can diagnose or we can identify carriers at early stage, long before disease strikes. So when we're talking about genetic variations at DNA level, where DNA is the blueprint, at RNA, like I said, it's like a manual, execution manual. So RNA tells us what is happening inside a cell at a given moment. Okay, so think of it in this way, that every cell has a same DNA, but not every cell expresses the same genes. Okay, and with help of let's say understanding the variations at RNA level, so I'm talking about the gene expression level, right? So it helps us to ask which genes are turned on or off, or which pathways are active or suppressed at that particular time frame. So, for example, let's say let's say in healthcare, so cancer versus normal tissue, right? So when we're comparing like gene expression changes at for those two diverse samples, let's say we might see inflammatory genes firing at full speed, or let's say immune pathways being switched off. So these differences in gene expression can explain why two patients with the same mutations have different outcomes. Okay, and again, at RNA level, it's not just about the amount of RNA or the gene expression. Sometimes, RNA is cut, stitched, or even fused in different ways, like a novel isoform of a protein, a gene fusion, or let's say completely a new transcript that we didn't even know existed. So these events can always be predicted from, I mean, it can't be predicted from DNA alone. So we can integrate one more layer of our study, transcriptome analysis, or based on RNA. So studying RNA gives us a live snapshot of the cellular activity, okay, beyond those static code of DNA. Right? So we have seen how DNA gives the blueprint and RNA tells us what is happening in real time. But ultimately, it's a protein that do actual work. So there are the proteins. They are the enzymes, the receptors, the scaffolds. The signaling messengers. So they are the real doers of the cell, or let's say they are very close to the phenotype. So this means if we really want to understand health and disease, we need to look at proteins, because that's where the action plays out. So how much of each protein is present at, let's say, at different conditions. Let's say we are talking about different diseases or treatment. If we understand that, it can completely shift protein abundance. So how are proteins interacting? A single mutation can change how a protein folds and whether it can bind to its, let's say, the partners. Okay, and this is why proteomics is a powerful complement to genomics and transcriptomics, or let's say to understand the variations at DNA and RNA level. So by integrating all three, we don't just see the blueprint, or let's say the plan. We see the entire machinery where action is happening, right? And how it breaks down in disease, right? So, but of course, we also have epigenetics or epigenomics. Like I've explained earlier, even if two people have the same DNA sequence, their genes may not behave the same way. And that's because of the epigenetic regulation or control here, which they regulates or regulates how genes will express, or let's say either they are switched on or off. So again, I will take an example of cancer. Right? So here we see many cases of hypermethylation of particular gene, or of that particular promoter, right? Specifically in those DNA repair genes. So the sequence itself is fine, okay, but the promoter is let's say chemically silenced, that methylation. So here the repair system fails, right? So an accumulation of mutations and tumor progression. So it helps us to understand how we can treat the patient. And then, of course, it's not just focusing on, let's say, DNA, RNA, and protein. So there are uh, uh, different sections as well, right? So to study these layers of complexities, so what we say, we have an umbrella of omics, under which we have genomics, like looking at your DNA blueprint. We have transcriptomics, checking which genes are switched on, okay, or off. We have proteomics, we are measuring the proteins that actually do the work. We have epigenomics, studying the switching on off of that control genes, how they are controlled. Okay, and of course there are different omes. Like for example, we have microbiome. We have specifically for metabolomics, we can focus on how the pathway works, right? So these are the complete study of these omics. We can call it as a multi-omics. And why I'm focusing on multi-omics? Because at individual level will help us to understand. So what the question we started with, right? Why two patients, or why two different plants, or even why two different individuals behave uniquely? Okay, and but studying genetic variation at particular one level will give a start point, but not, let's say, complete answer. Okay, and then studying these genetic variations at each level, integrating those data, is what we call it as multi-omics. And specifically for complex diseases like cancer, where one ohm or one omics will not give you complete picture. Multi-omics will play an important role, or rather it is playing an important role. Okay. Now, of course, now you might be wondering that how do we actually explore all of this layer of let's say biology in the lab. Okay, so I'm talking about the wet lab perspective. Okay. So is there any one particular technology that gives us insights into DNA mutations, RNA expression, or let's say even the gene regulation? Or can we decode the complexities of life using an unified approach? Okay, so answer is yes. So, and this is where I would like all of your attention towards this marvelous technique, next generation sequencing technology. Okay, so rather than NGS, let's say the sequencing technology, because they have evolved, right? So at DNA level, NGS allows us to identify variants like those SNPs, indels, CNVs, right? That helps us to identify mutations driving diseases, germline and somatic alterations, and also microbial identification. I mean, for the metagenomics part. Okay. NGS at RNA level helps us to explore those uh, dynamic gene expressions across different conditions, alternating splicing or isoforms or gene fusions. Right? It also helps us to identify those regulatory non-coding RNAs, right? Like the long coding RNAs or microRNAs, and also the transcriptomic signatures of healthy versus disease. Okay, at epigenetic level, NGS helps us to identify or to study the DNA methylation profile, okay, the chromatin accessibility or histone modification. So this is the unified technique. So in many ways, we can say that next generation sequencing is the common language across omics, or let's say for multi-omics. So we can add it like having a single universal scanner that can read the instruction manuals. Okay, so that can read our code. Okay, and that's why what I feel, whether you are a student of masters, I mean your PhD student or your postdoc or your professor working, right? Whether you're working in industry, whether you're working in research, whether you're studying cancer, infectious disease, or precision medicine, right? Or you're working in bioinformatics field, next generation keeps showing up, or it will keep showing up at every level. So now, I mean, like one of the when I started my career in NGS, right? So I thought that sequencing, right? So always thought that it is for research purpose only. Okay, because what we with the help of whole genome sequencing, we now know where to focus, where to target for particular disease. Right? But I would say now currently its impact actually has exploded across virtual every major industry. So next generation sequencing, or let's say will coin, I mean NGS actually is no longer just a discovery tool. So it's actually driving real world solutions. Right? But when I'm talking about real world solutions, means, for example, let's say we'll take the healthcare and diagnostic sector. Okay, so here NGS is helping to diagnose, to treat, and even prevent disease. Okay, so our idea is to have those core of personalized medicine. Okay, we have already always had that drive to have those personalized medicine. NGS is helping it. So let's say think about oncology. So mostly I'm talking about cancer or oncology because I'm working on that in that field. So with NGS, here we can now perform liquid biopsy. So when I'm talking about liquid biopsy, is like a simple blood can, let's say we can get a blood sample from a patient, and it can help us to capture those circulating tumor DNA or ctDNA, if you have heard. Okay. And when we're capturing those ctDNA, circulating tumor DNA, it allows us to detect cancer driving mutations, and that too non-invasively. So that offers the potential for early cancer detection and also monitoring treatment response. Okay. Of course, it's easy to say than to do. There are a lot of challenges, but at least, with these cutting edge technologies, we are moving forward to that dream of the personalized medicine. Okay. And it's not just in healthcare. Okay. So where in the world, where we are talking with the growing population and also changing climate, next generation sequencing is critical for ensuring a stable and let's say nutritious food supply. A fantastic example of this is in crop development. Now what scientists, it's not now, but what scientists are doing is they are sequencing the genome of countless varieties. Okay, let's say for example rice, wheat, bajra. Okay, so they are sequencing the entire genome, and after sequencing of that entire genome, it helps them to pinpoint the exact genetic traits, that allows some to, some of the variants to be, not variants, some of the varieties to be incredibly, let's say, particularly tolerant to drought or cold. Okay. And once they identify those genes, with help of breeders, they can develop new new varieties. Let's say, for example, drought tolerant variety of rice. Okay, but and this has been done, I mean, from ages, but with help of NGS, we can do it. We can fast track this process. Okay, that can help us to secure, a vital food source for billions of peoples for all over the world. And I would say that this technology is not only limited to humans and plants. Okay. NGS or next generation sequencing also plays an important role to understanding the life that is invisible to the naked eyes. Okay, so consider the health of our ocean. Okay, so using NGS or using the let's say a technique called metagenomics, researchers can take a single water sample and sequence all DNA within that water sample. Okay, so this gives them a complete snapshot of the marine microbiome. The bacteria, the virus, and fungi. What kind of organisms are there that that form the base of that aquatic food web. And this is very crucial for monitoring the effect of climate change, detecting pollution, understanding the overall health of the most vital ecosystem. And like I've mentioned earlier, okay, so NGS is not just used in academic settings or let's say research settings. Okay, so it is also applied over different industries. So one of the powerful application here is to understand the drug resistance. So in cancer therapy, for instance, tumor can evolve to resist treatment, okay, different treatment. So using NGS, and it's been done, we can track the clonal evolution in real time. So by sequencing tumor cells over the course of treatment, we can see which new mutations are emerging. Okay, we can understand why a drug is failing and make informed decisions, to either switch or continue with that particular drug or therapy. Okay? And then, of course, there is one more sector where NGS is applied. It's in criminal investigation. Okay, so we are all familiar with DNA fingerprinting. Yeah, most of you have studied in your either in your bachelor's or let's say even in 10th or 12th why DNA fingerprinting is important. Okay, but here next generation sequencing takes it to a new level. So a compelling example here is, let's say, wildlife forensics. Okay. So now, to combat the illegal wildlife trade, okay, what researchers or scientists are doing is they're using a technique called DNA barcoding. Okay. What they do is they can sequence DNA from, let's say, a particular item, let's say piece of leather product or ivory. Okay, and they can match it to specific endangered species and sometimes even pinpoint its geographic origin as well. Okay, so this provides the hard scientific evidence needed to prosecute those traffickers. Okay, and protect the vulnerable animal populations. So I mean, the bottom line here is that next generation sequencing is not a monolithic tool. Okay, it's not just for limited to research. Okay? It's a foundational technology that is reshaping entire industries. But the most critical thing to understand is that this revolution is not just about what we can do. Okay, it's about the staggering or almost unbelievable speed at which we can do now because of this evolution of this technology. Okay, I will give you a short example. Okay, modern Sanger sequencing method, right? So we all know that we have sequenced the human genome, and the modern Sanger sequencing method has played an important role here. Okay. Uh, it took like around 22 years to complete the entire genome and to fill the telomeres as well, more than that, right? So now let's say, for example, through this technique, okay, we can get around 700 base pair or around, we can sequence around 600 bases. Okay, if we are sequencing, let's say, a human genome, okay, on one instrument, one modern Sanger sequencing instrument, right? So how many bases we have? We have around 6 billion bases. I'm talking about deployed human genome, okay, and generally we need at least 30x coverage. But here, let's say if we are going with 7x coverage, okay, so we are covering 7 times. It will take at least 100 years on one machine. Okay, but of course, we had completed less than 100 years, right? The complete sequencing of genome. Why? Because we had set up the factories of those sequencers, and day and night the researchers were working, to stitch those bases, to align those bases. Fast forward now, okay, with help of let's say now the new age technologies. Okay, so this is a NovaSeq X series from Illumina. We can we can at least sequence. I'm not talking about analysis, but we can sequence the entire genome within 4 to 5 days or 3 to 4 days. Okay, of course, it will take some time for data analysis. That's at another level. Okay, but you can see how the shift of the technology has impacted, right? So when I said that it's not what we can do, It's about how fast we can do. That will matter. And this is where, NGS or let's say the new age technologies in sequencing, which have evolved so drastically, helps us. Okay, but I would like to quote here is that, with great powers comes a great risk of confusion as well, especially if we don't understand the methodology behind what we are doing. Okay, let's say, for example, we will take genomics. Okay, now in genomics, NGS isn't a single, let's say, recipe. Yeah. It's more like a different tools in toolkit. Okay, when we are studying genome, we have three important approaches: whole genome sequencing, whole exome sequencing, and targeted sequencing. Now, whole genome sequencing uses big picture. It captures everything. Okay, it's powerful, but also costly and data heavy. Whole exome sequencing, this frames the search, let's say, down to only those coding regions. Now, coding regions, we have, let's say, 1 to 2% of our genome, which is I mean, important for the, let's say, those codes, right? Where most known disease causing mutations live in those exomes, right? So in whole exome sequencing, we are focusing on those 1 to 2% of genome. Okay? And in targeted sequencing, here we get very specific. Okay? So zooming in on certain coordinates of genes or panels of genes. Right? So like in cancer, there are certain hotspots or drug response markers, which we already know. Right? So we can, I mean, targeted sequencing is used specific for diagnostic purposes. Right? So it is fast, cost effective, and especially practical in, in those clinical setups. Now, here's the key point. Okay, so key point is choosing between these approaches is not just a technical decision, but it can also make or break your study. Okay, so pick wrong one and you risk either drowning in those irrelevant data, or you can also miss the signals which you are searching for. Okay. And again, the challenge of sequencing one genome, I mean, let's say, of course, here when we're talking about genomics, we are focusing on one genome, right? So imagine sequencing many genome in the same sample, right? So again, it adds layer of, challenges. Let's say we want to analyze the microbiome of soil sample to understand its health. Right? We can't just use human reference genome here, right? So we have to choose a different strategy, such as 16S RNA sequencing or shotgun sequencing methods. So even before we start sequencing, we already have to make strategic choices, and which kind of approaches should I use, or whether should I go for targeted sequencing, how to do, right? And when I'm talking about how to do, so that actually brings me about the workflow of NGS. Okay, so let's say in your research objective or your work, you have decided which kind of approach I need to do. Now you have to execute it, right? So when we're talking about NGS workflow, irrespective of, let's say, the technology, okay, whether you're going for short read sequencing or whether you're going for long read sequencing, it can be divided into 4 aspects or 4 steps. Okay, and the lack of expertise here in any of the steps, it can derail the entire experiment. Okay, so let's say, for example, if we are talking about step one, okay, so you need to prepare the samples. Now, we had seen, right? We had seen different health sector. We have seen agriculture based, the microbiology or biotechnology industry, or even forensics. The samples what we get changes, differs, right? But it all starts in the step one. So there is a very different, I mean, famous saying that in NGS field is that garbage in, garbage out. So that is an absolute law of NGS. So if your initial DNA or RNA from the sample what you have isolated, right, is of poor quality, let's say, or poor quality or low quantity, or let's say contaminated, right? So no amount of fancy sequencing will save it. Okay. So this step, the extraction step, is deceptively simple, but is often the single biggest point of failure. And then the second step here, uh, once you have isolated the nucleic acids, either it can be DNA or RNA, you need to make it compatible to those particular sequencer. Okay? Because still, at least now, we don't have, up until now, we don't have the technology where we can just isolate the nucleic acids and we can directly just pass on to the sequencer, and it will get those sequences. So for example, in a cell, the DNA is, it's quite tightly packed. All those histones plays important role. If we untangle them, and if we stretch them, the length is around 6 feet of that in a single cell, the DNA, right? So we can't sequence those at in the entirety. Okay. Here, so to make it compatible with sequencer, in step two, what we do is we fragment the nucleic acids. Let's say we are talking about DNA. Okay. So what we do, we fragment the DNA, and there are certain processes where we make it compatible with sequencer. Let's say we are talking about short read sequencing. Let's say we are talking about Illumina technology. Okay, so there are certain type of adapters which are needed to, let's say, ligate or to add to those fragments of those template DNA. Okay, and of course, let's say a subtle mistake or the wrong choices of kits or methods, here it actually, in this step, it can introduce biases, that will corrupt your final data. And of course, then we have the main event, the sequencing itself. So once you have extracted nucleic acids, you have prepared the libraries. Now the prepared libraries is loaded onto the sequencer. Okay, here it is where it will read out millions or billions of DNA fragment. While this stage is highly automated, okay, but sometimes things may go wrong because there are a lot of technical aspects which we need to understand. Okay, so you need to know how to monitor the sequencers and assess the quality of those raw output. So congratulations, so we have done the sequencing, and finally we reach at the last stage. That is the most challenging part again, data analysis. Okay. So this is where the raw data what we generally we are getting, we are making some sense or a sense of those data, right? So in analysis step also there are certain steps, let's say primary, secondary, and tertiary analysis. So primary analysis is where those the reads, actually they're converted into the bases, A, G, C, T. So there are base calling is done. Secondary analysis is where, let's say, in case if you have reference genome, you're aligning. Before aligning, of course, there is demultiplexing. If you're using adapters, you need to trim them. Quality check is there. Okay, once that is done, once let's say we are aligning with reference genome in the secondary analysis, then in the tertiary analysis is where you're making the sense of those data. You're making the sense of those AGCTs. This is where you are generating those plots, those heat maps, identifying genes in specific pathways, right? And this actually cumulative, you know, uh, here the workflow or NGS technique, right? This helps us to answer few of the questions, not entirely. The what actually we started. So here you can see the journey from a biological sample to an let's say into an actionable insight. It is not linear at all. Okay, so it is layered. Okay, there are technical aspects. Okay, there are experimental aspects, logical aspects, what you need to study, do, and it's often overwhelming. Okay. What I feel, working in almost a decade now here. So the real chaos in NGS isn't the sequencer or the technology itself. It is the dozens of decision that surrounds it. Okay, so which method to choose, how to prep your samples, how to design your experiment, and more importantly, how to make sure you're interpreting it all correctly. Okay, so again, like mastering NGS is not just about clicking buttons or just following a kit or following a protocol. It's all about understanding let's say the why, the how, and what if at each step. So this is where actually with help of OmicsLogic, what we did is we have designed a particular basic course in NGS. So there are a lot of challenges to understand, and what we have done is categorically we have yeah. So what we have done is we have divided into certain aspects, to reduce or to help you all to navigate from those complexities, to keep you updated what actually is going on and how you can leverage from this technology. Okay, so this is where we are building a foundation in NGS, so that can help you to climb your ladders for your particular research objective. So here in week 1, what we are doing is we are introducing NGS. Okay, so what we have planned is that we'll start with the big picture, right? I mean, we'll cut through the jargons to understand how different sequencing platform works and what the future holds. Then in the week 2, the we will discuss about, or we'll learn about the NGS workflow and sample preparation. Okay, so what actually the workflow what we have just discussed now. So we'll start with how actually samples are prepared, why quality control is important. You will learn the do's and don'ts that can make or break your experiment before it even begins, right? In the third week, we are will dive into the library preparation techniques. We'll dive into both the DNAs and RNAs, right? Which is a core molecular biology part of the wet lab process. Okay. So we'll explore different methods for preparing your samples, for sequencing, whether you should work with DNA or RNA, or why you should work with DNA or RNA, the experimental setups. Okay. And in week 4, what we have planned is advanced library prep. And of course, in that we have also added quality control. In the preceding weeks, we'll discuss some strategies where we'll bring everything together. What we have studied, what we have understood, to focus on how to design the experiments. Okay, and we will also have some real world case studies. Okay. Now, of course, it's not that. We are endorsing a particular program here, right? So the main objective behind designing this course, of course, is creating awareness so that you know how to leverage this technique or how to leverage this technology. Okay. Of course, this will give you a solid practical foundation in NGS. Uh, but my personal objective is something deeper, right? So it's like to instigate curiosity, what I have learned, okay, in the process of doing NGS, okay, and to help you all to ask better questions about the biology, technology, and then what possibilities actually it that lies in between. Okay, so my goal here is simple: to help you not just learn NGS, but also to leverage it, to innovate, to research smarter, and also to stay future ready. Okay, so hopefully you have learned something new in this webinar, and hopefully you are taking something from our discussion. Thank you very much for your time. Yeah, Shri Gauri.
Yes, thank you so much, sir, for the very insightful presentation. And we also got to learn a lot about NGS, and I'm sure a lot of the participants are also finding it insightful. So, just in continuation, before we move on to the Q&A, before we take up any questions from the participants, just want to build upon the topic that Dr. Rahul was covering about the NGS course that we are launching. So we have a detailed brochure with all of the information about the course, like what all topics are covered, and also some of the subtopics. When are the sessions scheduled? What will be the duration? What are the various resources that you'll be getting access to under this coursework? What are the various learning outcomes? Who can attend, and so on. So in case you do not have access to this brochure, we will be sending it out via email along with the recording from today's webinar. So just keep an eye on your inbox. We'll be sending it out so that you can have a look into it and learn more about the topics that were covered during the webinar by Dr. Rahul. Okay, and for those participants who might be interested to enroll, we have a page ready for this program. I'll share the link in the chat box so that in case participants would like to directly enroll, they could do so. So I've shared the link in the chat box. This is the website, and you can scroll down below to have a look at the once again the curriculum that is being covered throughout the coursework. And as you scroll down below, you'll be able to get to the page where you can proceed with the checkout option. And the pricing is displayed. You can change the pricing based on your location, that is in USD, INR, and so on. Okay. And then just click on Buy Now. And as promised, for those participants who have attended today's webinar, they will be getting a exclusive discount option. So we'll share the discount code in the chat box. That is NGS35. Thank you so much, Bhavna. So you can just apply this code at the checkout option, NGS35. Just click on apply, and the coupon is successfully applied, and you can just enter basic details of yours like contact details and so on, and you can select the payment option that you would like to proceed with for purchasing the program, that is PayPal or Razorpay. And this coupon code will be active till 7th of September. So please make sure that you enroll before that the coupon becomes expired. Okay. And in case you do not find the option that is suitable at your location to proceed with the program, you can just email us at communications@omicslogic.com, and one of our team representatives will get in touch with you and assist you with further enrollment options. Okay. So that was just all about that we wanted to cover about the course enrollment option as well. Shubham has also shared in the chat box some of the contact details of our team members. So if you would like to have a discussion with our team to understand more about the coursework, you can get in touch with them. They're also available on WhatsApp, so you can just drop a message to them there as well. Okay, so is there any other questions from the participants? I can see, okay, from Anamika is 35% discount. Yes, it will be 35% discount and it will be conducted online. There will be sessions conducted, one session per week, so that you can catch up in case you're not able to catch up with the recording and you would like to catch up with the session. There are recordings also provided for you, and you can view it up to 1 month so that in case you would like to revisit some of the topics, you can do. And there were also a few queries about the feedback link. So in case you would like to share any feedback, you can email us once again at communications. And of course, we'll always look forward to hearing about how you find the webinar, about the coursework, anything, any other topics that you would like us to cover in the upcoming webinar or programs. We'll be happy to look into that. Okay, so Sagnik is asking, is the certificate provided? Yes, for the course, once you complete the coursework, you will be getting access to the certificate that you can showcase on your resume or on your LinkedIn profile. I can see Gopalan has raised his hand. Would you like to ask any query, Gopalan?
Yeah, absolutely. It's more related to technical. So it's like I just started out with my literature review. I'm sort of about 1 year into the literature review, and I'm like at the process of actually zeroing on my topics. So there are some interesting papers that I came across. There was this paper. This actually said if there are genetic mutations, there is a possibility that we could actually make use of the second technique, which I remember, not the whole genome sequencing, it was exome sequencing, if I remember, or targeted sequencing, which they made use of. And they actually extracted a part of that DNA, and they actually extracted the DNA of a bacteriophage, and they actually ensured that the sections of the bacteriophage are such that they are actually compatible with the original genome of the person. So this was mostly ex vivo, actually not in vivo. They had actually extracted the cell and they had extracted the DNA, and they had run this process actually through an experimental affair, and it's still under testing actually. The query that I have here is, see, the treatment is specifically for a genetic disorder. So if, let us say, what is the guarantee that there would not be a mutation if we were to, inherently a bacteriophage has a different form of genome sequencing, although there is some sense of compatibility, there is bound to be certain differences in the genome sequencing of a bacteriophage and a human. So that's
Yeah,
exactly how we are actually able to have a difference between a microbe and a human being. There are different
yes, yes.
levels of sophistications, right?
Correct. Yeah. So it is. I mean, when we are talking about mutations, let's say at DNA level, we always say it's they are static. Okay, so of course they. I mean, when we are talking mutations, they are more spontaneous. It will happen. So the question here, it's not about let's say whether it is right or wrong. The question here is how we can identify it.
Hmm.
Okay, so yeah, so this is where actually because the question what you ask is quite scientific, so it will
Yes, sir.
require a
It's
lot
like,
of research and improve. Yeah,
yes, sir.
go ahead, please.
This is the first part of the question. There is just to inform you, there is a sort of a spin-off question from here. So you please continue, and I'll ask the next question there.
No, please go ahead. Ask the second
No.
question.
Please continue answering this one. So we'll
Hmm.
complete the technical part, and then we'll move on to
So
sort
the
of a lead up
yeah.
question from here.
So what you said was, I mean, you were asking if I understood your question is, of course, at cellular level or genetic level, we'll have those different mutations, and we are not able to pinpoint certain things technically, to understand how to know the approaches what we have discussed. So I've just given a hint. So we were discussing about whole exome and targeted sequencing. Now, once we know which variations or where the coordinates of those variations are, we can design certain panels. Okay. Now, what, let's say for particular part, it's all validated variations. We are just focusing on that specific coordinates. We are sequencing them. Now, in sequencing, one thing you should always remember is the depth of sequencing, the coverage of sequencing. Okay, so it's not that, because we are talking about those single nucleotide variations, and anyways, with help of the NGS, we are there might be biasness. Okay, biasness for that particular. If you're done PCR, right? So there might be a biasness, and for that, what we do is we go in depth. So when we are going for targeted sequencing, we are going at more than like, there are instances where we are going more than 1000x coverage. We are checking. So that gives us a precision about or confidence about that variation.
Okay.
Yeah. Your second question?
The lead up question is like the second part is would this not lead to some form of bioethical concerns here? Looking at from the perspective of a policy making or a person
No,
or a
let's
medical person or from the perspective of a government. So would it not
bioethical.
raise bioethical concerns?
Can you give an example?
See, the person actually comes to us for a treatment under the impression or probably under the trust that we would be able to cure the person's genetically induced disease or disease that is induced by mutation by birth. Fine. And we treat the person, and we also do that under the trust that we would be able to cure the patient. But unfortunately, what happens as a consequence is that there are two ways to look at it. One is you actually cure the person, and you are actually gaining access to the person's genetical data here. So you have to ensure and to ensure that you're actually able to address the future concerns of the very same patient, you need to have the personal data of the person, which
No.
might
I would
actually
just like to ask you one
yeah.
question. Sorry to interrupt,
Yeah.
but which kind of treatment are you discussing? Can you give an example?
It's specifically related to genetic diseases that come.
Like?
Let's say if you take there is this diabetes that comes by birth for children, and there are certain diseases like let's say if you have some form of mutations due to which your color of the iris changes and you want to make it back to normal, like black
No.
due to which,
So
or some form of blindness that comes out
yeah. So
by
I
birth.
would like to draw a thin line here. Okay, it's a
Okay.
very good point. It's a very good question you have raised here. Okay. Now let's say, who takes the decision here of treating?
The doctors and the
Yes,
bioengineers
exactly.
together.
And who and the doctors takes the decision, right? So there are specific guidelines. So when we are talking about NGS, what generally we do is we give the data to them. Okay. Now data is where we are taking care of. Okay, we are taking care of that. It should not be contaminated. It should be precise. Variations are there, and after that, the decision is of doctors. Now, how doctors also take precise, let's say, decision is based on there are certain guidelines. So you're correct. So it might be deceiving, right? So it depends on those regulatory guidelines, and there are, let's say we are talking about cancer. Okay, now there are FDA approved drugs which are correlating with those certain variations, and lot of studies have been done in backend, and then only they have been approved. Okay, so it's like, it's like internet, or let's say it's like now we can say chat GPT, how you are using it. Technology is there for you, right? It depends on specifically you. This is a very, very valid question, and all those who are attending, we should also think in that same way, right? But here, like I said, technology is not responsible for that part. If I'm answering your question, right? So it's not
I
the
get the
technology.
point. It's like
Yeah.
how human is making oneself responsible, or rather handling it in more responsible ways, what you're trying to put across.
That
The
yes,
responsibility will rest with the engineers and doctors so that they're able to do it ethically, is what you're trying to put across.
correct,
Correct?
exactly. Yeah, exactly. And thank
One
you
more
for
lead up
bringing this
question, I'm
question.
sorry. It's related to the contaminated specimens.
Hmm?
Wouldn't the contaminated specimens also give us a different perspective of how things would transpire
Yeah,
under
true,
such conditions?
exactly. Exactly. So let's say when you're doing transcriptome analysis, okay, so your contaminant is your DNA. When you're doing DNA, when we are working on whole genome sequencing, your contaminant is RNA. When you're working with certain, let's say we are talking about healthcare sector, okay. When you're working with, let's say lung cancer, okay, and there are certain type of oral cancers samples are there. So contaminants are oral cancer. So this is where, if you remember when I was discussing about the workflow, the sample extraction. So I had used those, that specific word, that garbage in and garbage out. So exactly. So this is where we need to take care, and it's our responsibility as a researcher, to feed the sequencer the correct nucleic acids without contamination. Yes, of course, yes, it does. Sequencer, the process it will follow.
Okay.
It's us who need to take care.
No, the reason I put this question was when I did get some contaminated specimens, and when I was working in the lab, and I got some anomalous results actually.
Yeah.
So I wanted to just get a heads up whether I could make use of such data so that I'm able to give a different perspective to the
So
research that I'm.
which kind of objective you had? So
Uh,
which
the
approach you did?
objective is basically to try out, uh, specifically what I'm trying out here is the target area here is basically with respect to two things. One is genetically induced disease, that is by birth. That is one part of the objective, and the other part of the objective that I am using now is how it leads up to cancer,
Okay. No,
because
your
the later
the
part is
objective is.
actually dealing with the tumor and cancer cells, and
Mm,
the initial part is actually dealing with the genetics basically.
okay.
So how the contamination in the genetics could probably lead to you
Yeah.
know uh, cancerous cells or carcinogenic exposure to carcinogenic stuff can lead to cancer due to specific genetics, is what
Mm,
I'm trying to actually look through, and I'm trying to build up on that area. So since I'm just with my first year, so I'm just
mm,
able to give you a very, very vague picture of what I'm trying to do. I'm still
Mhm.
actually in the process of refining my literature survey actually.
Okay.
So,
Anyways,
Hi,
this is quite
sorry to
sensitive.
interrupt. So sorry to interrupt. Just wanted
yeah,
to say that the meeting link will be closing in about 2 to 3 minutes.
Okay.
In
So
case other
what
participants have queries,
yeah,
you can email us.
what we can do is, of course, now time is also done. So whether you have any questions, you can just Shubhangana, you can just add the email ID, and
yeah,
then we can
absolutely. That will
address
be
those
very
questions.
useful, sir. So if
Yeah.
you could, share the email ID, I could, not the Omics mail ID. I probably
So
wish to directly interact with Dr. Rahul
sure, sure,
Ramekar.
sure. Yeah.
That'd be fine.
So
Okay.
you can also pass on the queries to communications ID and we'll be passing it to Dr. Rahul. Okay.
Oh,
Exactly.
it will be like via. Okay, I get it. Or I already sent you a LinkedIn request. So could we interact over LinkedIn? Would that also be fine?
Yeah. We'll do that.
Yeah, absolutely. Sure. Thank you.
Okay.
Thank you.
Thank you very much.
Thank you.
Yeah, even Sagnik Ghosh is asking how to detect transition and transversion. So those are most kind of basic biology questions, and it will be very difficult to answer here in this webinar. So Shrigauri, I mean, what we can do is whether if they have any basic questions, please do forward me. I will try to answer them,
Yes, sir.
and we can end the webinar here now.
Yes, sir. Okay, sir. Thank you so much, everyone, for joining. Just as a quick recap, I'm just sharing the coupon code in the chat box if anyone is interested to enroll in the course. And the coupon code will be expiring by 7th of September. So please make sure to enroll before that. Thank you so much, everyone, for joining in for today's webinar. And we look forward to seeing you in some of the upcoming workshops or coursework. Thank you so much, Dr. Rahul, and thank you, everyone.
Thank you. Thank you very much for your time.
Thank
Hope
you.
you have learned something new. Yeah. Thank you. Bye bye.
Thank
Have a
you.
good night.